Today, we are here with Dr. J.N. Verma. He is the founder, Chief Scientific Officer and Managing Director of Lifecare Innovations Private Limited. So, hello Dr. Verma. We understand that you are an academician. So, what was your inspiration and motivation for starting Lifecare Innovations Private Limited?
I am a liposome technologist. I have been working on liposome technology since 1977. It has been more than 40 years now. And I used to work in United States where I started to use this technology for the product development. I was working on the malaria vaccine development, liposomal malaria vaccine development. When I came to India in 1990, the Indian government was grappling with this issue of making India self-reliant for liposomal amphotericinb. It's a critical care drug, but it was being imported at that point in time and it was exorbitantly expensive. So the government of India has started a mission to develop and manufacture liposomal amphotericinb. I was approached that if we can further work on this technology which was in the academic circles being developed and also manufacture this thing. To take up this, Lifecare Innovations was formed in the year 2000, and since then we have been working. In 2003 the product was launched by the Honorable Vice President of India on Technology Day 2003, and so this mission of making India self-reliant for this very expensive critical care drug was enough motivation and inspiration. Yeah.
So, what is your area of research interest and what are the products that you have innovated at Lifecare Innovations?
Komal, as I said that we started this company with making a drug which is used for treatment of systemic fungal infections and also for Kala Azar. So the company's focus first was on these two areas, but then we started to look at other possibilities and we developed different formulations of liposomal amphotericinb. We have a topical liposomal amphotericinb gel which has many applications for treatment of topical fungal infections. Then we had to focus. We try to use liposome technology to address other unmet medical needs, and we got into the psoriasis, and we have developed 3 products for treatment of psoriasis. We have a liposomal dithranol, and we have a special formulations of coal tar, which is a lipoidal formulation of coal tar. The 2 formulations. And very soon we are going to launch another new drug. In time to come, we will make that announcement too.
As we all know that there are many liposomal amphotericinb drugs available in the current market. So, how is your product, your liposomal amphotericinb, different from the rest?
Okay, it's not only that liposomal amphotericinb are available. There are many formulations of amphotericinb that have been developed. Some stayed and some have even been withdrawn. Why this is happening and what are the important issues involved in amphotericinb IV formulations? Amphotericinb has certain merits. These are matchless merits, but it also has some problems which are very difficult to deal with. Amphotericinb is the most broad spectrum antifungal. This drug has been in use since 1959, and in more than 60 years there is no drug resistance. It's a remarkable thing. The drug is a very potent drug. Of all the antifungal drug, amphotericinb is the most potent. But then there is a problem. The problem is that amphotericinb is extremely nephrotoxic, and its nephrotoxicity can lead to renal failure. Now, when the drug was discovered, to formulate it was very, very difficult. And the first formulation which came, which is known as conventional amphotericin B, has been used world over, is a deoxycholate suspension. This drug has a toxicity which affects at least two-thirds of the patients who use this drug. In the absence of any better option, there was effort to improve the clinical performance. So work on the formulations. In 1964, a new technology came. It's called Liposome Technology. It originated at Cambridge University in the laboratory of one Dr. Alec Bangham, and this liposome technology was actually originally meant to make artificial cell-like structures. So liposomes are like artificial cells made up of phospholipids, and this was meant to study the structure and function of biological membranes. After a few years, some of the scientists working in his lab thought of applying this technology for the delivery of bioactive substances. Bioactive substances means the drugs and vaccines. And one of the people who worked with Alec Bangham was Colonel Alving of Walter Reed Army Institute of Research. That's the place where I started to work on the product development. So Colonel Alving was working on liposomal malaria vaccine, and I was working in his laboratory on this liposomal malaria vaccine, and we were the first one to show that this liposomal vaccines give you cellular immunity also in addition to the humoral immunity. So from here the product development started. The first product using the technology, the first drug which was developed was liposomal amphotericin B. Now different formulations are identified by different names. So after the conventional amphotericin B, the next product was amphotericin B lipid complex. The third product was amphotericin B colloidal dispersion. The fourth product was liposomal amphotericin B, which is prepared in sucrose and dextrose. And the fourth product, our product, is liposomal amphotericin B in saline. These are evolving formulations of amphotericin B, and they all have different dose, different efficacy, different MIC, and different nephrotoxicity. Since our product is the only product which is made in saline, it is the most nephrosafe. More than 300,000 infusions have been given, and no nephrotoxicity is reported from this product. In addition to that, it has a unique composition which has increased its efficacy. So even at a lower dose, the efficacy is higher. So since you have to use a lower dose, it becomes more economical also. So it is a drug which is the most effective, which is most nephrosafe, and which is also most economical dose. And now, so that is how all these products are different from each other. Now you can see, I should tell you here that amphotericin B colloidal dispersion has been withdrawn from the market. It has been withdrawn because it was not giving the expected results. No, it was not a nephrosafe drug. It has very poor efficacy, so it has been withdrawn from the market. Similarly, there was a product which was a copy of the liposomal amphotericin B, I told you, in Argentina. That has also been withdrawn because although it had same composition as the liposomal amphotericin B, which was the first product, but despite having the same composition, it doesn't have the same efficacy and same nephrosafety. Okay, so this is the difference. I hope I have been able to.
That's good to know, sir. I wanted to ask in relation to the current scenario. As we all know that there is an ongoing medical emergency of mucormycosis, commonly called black fungus. So how effective is Fungizome in treating the patients of mucormycosis?
in case of COVID patients, generally, so far, whatever reports have come to the press or in the electronic media, it is seen that it is common in the patients who are diabetics and who are on the corticosteroids, who are on the steroids. So the mucor infection, even before COVID, is common. The infection itself is rare, but among the patients who have this problem of mucor infection, most of them are diabetics. And this infection grows very fast. So you need a very potent drug, and you need a drug which, if it is free of sugar, that would be best suited. Now, as I told you earlier, Fungizome is free of any sugar. We don't use any sugar. It is made in saline, so it is suitable for them. The MIC of Fungizome is much lower, which means it's very, very high efficacy. So it has two important features, two important properties: high efficacy and no toxicity. And that's why in case of post-COVID mucormycosis, it has a great role to play.
I just hope we save more and more patients from mucormycosis with the best treatment possible. I hope it's Fungizome. Thank you so much, Dr. Verma, for sharing your experiences and your knowledge about the particular product. And thank you.